Hi, everyone. Welcome back. I hope everyone had a nice lunch and some time to socialize with colleagues. And I hope some were able to attend the Mercy Ships lunch and the professional coaching sessions. My name is Emily Partridge. I am a general and fetal surgeon at Children's Hospital of Philadelphia, and my co-moderator is Dr. Stephanie Brearley, and she is a pediatric surgery research fellow at Phoenix Children's Hospital. This section is our second session on basic science, and it's my honor to introduce our first presenter, Dr. Yi Xiong, who will be presenting intestinal epithelium dysregulation in necrotizing enterocolitis.
So good afternoon, everyone. I'm Yi Xiong. Thanks for the opportunity to give me the chance to speak here. So I'm a pediatric surgeon, residency in training, and this is the program, great cooperation between China and Canada. So the necrotizing enterocolitis, the NEC in short, is a severe disease in preterm baby. There are many factors contribute to the NEC, and our lab mainly focus on the stem cells. So in this study, we aim to establish a multi-omic atlas using the mouse NEC model through the single-cell sequencing. Briefly, we perform single nucleate TAC-seq and single RNA-seq in mouse NEC model to provide a multi-omic reference map. Through the single nuclear RNA-seq, we identify the major cell types include endothelial, epithelial, fibroblast, immune, neural, and smooth muscle cells. And the epithelial cell mainly composed of is the largest population, and we found the epithelial cell is decreased in NEC. In the epithelial cell types, we identified the subpopulations include enterocytes, goblet cells, enteroendocrine cells, and others. There are some cell types such as enterocytes, stem, the TA cells decrease in NEC, while others such as goblet cells, enteroendocrine cells increase in NEC. And we performed a single nucleate TAC-seq, which provides additional regulatory information, the epigenetic change in NEC. For example, we identified differentially accessible chromatin regions in different cell types, and we performed a motif analysis. We identified the transcription factors such as MAD2 or 3 highly enriched in epithelial cells. We performed cell cycle analysis, and we found that the proliferation status in TA and stem is decreased, which was further confirmed by the Cassidy-7 staining. We know that villus blunting is a major feature in NEC, as shown in HV staining, and the V-line length is decreased in NEC. We perform a gene signature analysis, and we divide the enterocytes into the top, as shown in red color, and the bottom one as shown in blue color. The top enterocyte is more differentiated, and we found that in NEC, the differentiated enterocyte is decreased. Next, we integrate the single nuclear RNA-seq and ATAC-seq, and we computationally pair the cells from different modality, and we perform the trajectory analysis. We identified two trajectories. The first one is absorptive branch, and the second is secretory branch. The absorptive branch mainly include enterocytes, and the secretory branch mainly include the goblet cell and the enteroendocrine cells. And in the absorptive branch and in the secretory branch, we identified the transcription factors such as ASL2 highly expressed at early pseudotype, and FOXG2/3 highly expressed at later pseudotype, which may help to maintain the progenitor state or help the goblet cell differentiation. And we performed differential gene expression analysis along the pseudotype, and we identified the DEGs such as AXIN2, LGR5, CASP3, which related to stem cell pathways and cell death proliferation, which may help to explain the impaired pathways in NEC. And next, we performed the hotspot analysis to identify important gene function models. We identified model 1 to 14, which have different cell functions and the different modalities enriched in different cell types. Here we give one example. We found model 6 is decreased in NAC, and as we know that the internal size is decreased in NAC, and this model highly enriched the internal size, and this model 6 is related to lipid metabolism. In summary, in this multi-omic analysis, we found there alterations in epithelial cells in NAC, such as reduced intestine stem cell proliferation, reduced internal cell maturation and differentiation, and we performed the trajectory analysis to identify important transcription factors, motif gene change along the pseudotype, and we found the important gene modules. So here we complete the multi-omic atlas of epithelial cell changes in NAC, and we hope through our atlas we can mine some potential targets to revive the stem cell in NAC, which may have translational significance. I would like to thank for the support from Dr. Agustino Purol and all the lab members and the Capital Center for Children's Health. Thanks for listening.
We have time for 1 or 2 questions, if there are any from the audience. Yes.
Since nobody's standing to ask, I'm Casper Wong from Sick Kids, but I've never seen this data before, so I'm not a plant. This is very fascinating work, but I'm curious, one of the things that NEC is known, or it's been described with NEC, is that there's a impaired barrier mechanism to protect against the invasion of bacteria into the wall of the intestine. I'm curious with the secretory pathway if there are any signaling pathways that would suggest an impairment in the production of like the mucus barrier of the intestine or anything like that. Have you looked at it in that fashion to try and identify in the very trajectory pathways for your epithelial cells specific altered signaling pathways that are consistent with previously observed findings in NEC?
Yes, thanks for your question. The first is that so there's some in previous study there's some phenotype as indicated like actually standing like the actually something so nobody comprehensive understand this disease using the single cell seq. So our study is the first one to do the multi-omic atlas. And for your like the barrier, like bacteria, I think this is very important in NAC, and we show that, for example, we describe the change in the goblet cell, so which is related to the mucus secretion, and we are cooperation with the expertise in this part to further dive into this direction. Yeah.
I'm Rode. I'm the junior fellow at UAB.
Yeah.
I guess my question, you kind of touched on it in your future directions, but are you able to use any of the markers that you showed in your, kind of neck or not to detect, like for early detection of neck and like PCR samples, and also the other half of the question is, are you able to, like, you touched on like differentiation or de-differentiation, are you able to, drive certain cells to prevent, potentially neck in the future?
Yeah, thanks for your question. I think for early detection of neck, maybe you use urine or blood is a good way. So for us, we just collect the intestine tissue. So this help us to understand the mechanism in neck. Yeah, thank you.
Thank you very much.
Yeah, thank you.
Our next presentation is titled Untargeted Metabolomics reveals distinct metabolome changes in the gut-liver axis after massive small bowel resection. Presenting this is Dr. Daniel Alligood, a postdoctorate research fellow at Washington University in St. Louis.
Hello, everyone. My name is Daniel Alligood. I'd like to thank APSA for the opportunity to speak. I have no disclosures. However, I want to take a moment to first acknowledge my mentor, Dr. Brad Warner, who passed away last month. I know his absence is felt in this room, but his mentorship and sponsorship will stay with me forever. So I continue this presentation in his honor. So why is this research important? Intestinal failure-associated liver disease is a highly morbid condition in patients with short gut syndrome. It's believed the etiology is multifactorial, and by understanding the mechanisms of injury, we can maybe develop novel therapeutics to help these patients. Now, I think we all know the patient population that we're thinking of. In pediatric surgery, we're thinking of an infant that has neck, goes to the OR room with multiple small bowel resections, and ultimately gets short gut syndrome. And after that is a critical time where there's multiple extraintestinal and intraintestinal insults that can contribute to the development of IFALD. Extraintestinal insults can include perinatal nutrition, prematurity, recurrent sepsis. However, intraintestinal insults such as poor bowel adaptation, dysbiosis, permeability can also affect the development of IFALD. As such, the Warner Lab has dedicated the past 2 decades of using the parental independent mirroring model of IFALD that allows us to directly interrogate those intestinal effects on the development of IFALD. Here you can see a picture of the diagram of our surgery where we perform a 75% small bowel resection where a majority of the jejunum is resected and the proximal jejunum is anastomosed to the distal ileum. Importantly, there's 2 things I want to note from our prior work that we have published. In the SBR model, we have noticed that there's an increased intestinal permeability. We figured this out using fixed-dextrin assays, kinetic assays, and you can see these top 2 lines in the blue and red. The SBR condition has increased mucosal permeability compared to non-op and control. And the second interesting finding that we have published is that using immunofluorescence, we've looked at the lymphatic structure, which is the red protein here in this immunofluorescent stain. Hopefully you can see it. We've basically noticed that the SBR condition has decreased mucosal lymphatic structures. So these two findings prompted the question, do changes in intestinal permeability and lymphatic structure affect the transport of metabolites to the liver? And to answer this question, we decided to use a go fishing experiment doing untargeted metabolomics on a subset of our mice. And this cohort is a female cohort with 7 mice that underwent 75% small bowel resection and 6 mice that underwent sham resection. They were fed a liquid enteral diet for 10 weeks, and we measured liver injury by histological analysis and serum AST and ALT, and like I said, using untargeted metabolomics. So here I'm showing the serum AST and ALT values, and you can see here in red that the SBR condition has increased transaminases in both AST and ALT compared to the sham. And here I'm also showing representative histological images. Here in the H&E stains, I have circled various elements of steatohepatitis that's present in the SBR that's not present in the sham. And most demonstratively, you can probably assess that visually, you can see that there's more increased collagen deposition on the serous red staining in the SBR condition. Now, turning our attention to the metabolome of the portal vein serum, you can see first in this PCA plot on the left that the SBR condition in red compared to the sham condition in green have distinct metabolomes. And we can perform a differential abundance analysis, and here it's visualized in this volcano plot on the right. And you can see on the right side where the SBR in red, there's about 100 distinct metabolites that are uniquely associated with SBR, and about 122 in blue that are associated with sham. Now, the first thing we wanted to do was kind of get a sense of what are these metabolites. So we can classify them based on their structural classification. And here in the SBR condition, the first thing you can probably see that a majority of the recovered metabolites can be classified as fatty acyls, as compared to this is 30% as compared to the sham condition. Now, we can further subclassify these fatty acyls based on their fatty acyl chain length. And what you can most strikingly see that most of these metabolites, these fatty acyls, are long-chain fatty acids, and they're mostly in the SBR condition. And this is interesting because long-chain fatty acids are classically thought to go through the chylomicron lymphatic system instead of directly through the portal system. And then we can also look at individual metabolites. Here I'm just showing you a smattering of the top 10 differentially abundant metabolites in the SBR condition. And there's two things that kind of stick out to us. Here on the left side in orange, we have two derivatives of P-cresol. This is a specific metabolite of tyrosine metabolism that can only be produced by the intestinal microbiota. And we see that this is a very signature of the SBR condition. And then simultaneously here in blue, we see a lot of long-chain fatty acids that are present. So in a proof of concept experiment, we wanted to see, are these metabolites biologically active? Could they be contributing to hepatocyte injury? So in a very crude and simple study, we basically bathe HepG2 cells with these particular metabolites, P-cresol and its derivatives, as well as two fatty acyls, dihome and ethylpulminolate. And you can see, we measured cytotoxicity by LDH release. And as you can see, compared to the control, which is DMSO, we have proof of concept that at least at some concentrations, these metabolites can cause hepatocyte injury. However, this is not we don't know if this is reflective in vivo. So probably like any good experiment, this raises more questions than answers. But I think what this is suggesting, and my hypothesis, is that SBR is resulting in a diversion of fatty acids through the portal vein system. We hope to use a PET tracer with a radiolabel isotope of a long-chain fatty acid to dynamically watch to see the kinetics of the metabolism of these long-chain fatty acids, to see if they go to the portal first or the thoracic duct. And then secondly, the presence of these bacterial metabolites suggests there are some microbial changes in the SBR condition, and they may be contributing to toxin production and ultimate hepatocyte injury. So we hope to do shotgun studies to evaluate the metagenomic profiles. So I want to thank all the funders of this research, the Metabolic Technology Access Center who helped us, and all the members of the Warner Lab, and especially all the pediatric surgery group at WashU who's been very supportive during this time. And I'll take any questions now.
Thank you so much. In the interest of time, we'll take just one question.
Hi, Byerly, UAB.
Hi.
My condolences to you and your entire group and to a whole pediatric surgery family.
Thank
This
you.
was a very beautifully presented study. My question is, why did you utilize HepG2 cells, which are cancer cells? And in that utilization, can you take maybe some of the questions or answers that you saw from your data? And is this applicable to HCC?
So that's a very good question. So HepG2 cells was kind of we had these in the lab. We wanted to try it. We plan to isolate hepatocytes from mice to really interrogate it a little bit further. We have collaborated with some of the transplant surgeons who do HCC transplants, and we hope to liberalize our understanding of liver injury and seeing if there are similarities and differences between IFALD and HCC or MAFLD, et cetera. So hopefully we can have that presented in future studies.
Thank you so much. Our next speaker is Dr. Nicolas Vinny, and his presentation is entitled Cytokine-Driven Pro-Fibrotic Response to Mechanical Stress in Vitro in Primary Fetal Esophageal Fibroblast Cells.
That's the, yeah, okay,
The mouse.
sorry. Good afternoon, and thank you for the opportunity to present our work. We have no disclosures. Long gap esophageal atresia remains a challenge for pediatric surgeons because of the distance between both ends and because of the high rate of strictures. These strictures have been correlated to the tension applied on the esophagus. Fibroblasts are the main actors in the fibrotic process, and they have been shown to be mechanosensitive. We aim to understand the mechanisms driving esophageal fibroblasts activation when stretched. 3 different cell lines will be used. First, the FEF3 cells, which are primary fetal esophageal fibroblasts, and patient-derived fibroblasts either from EA patients, referred to as PDF-EA, and non-EA patients, referred to as PDF non-EA. First, we seeded and cultured FEF3 cells on a polycaprolactin scaffold, which mimics the extracellular matrix structure, and placed the cells in a bioreactor designed to apply tension on the cells. The cells were then harvested, and RNA sequencing was performed to compare gene expression between stretched and non-stretched FEF3 cells. This allowed us to identify 5 upregulated cytokines expressed in stretched FEF3 cells. Of these 5 cytokines, we decided to focus on IL-1 beta, which has been implicated in fibrotic processes in organs such as the lungs, the heart, or the liver, and has also a potential role in the response to gastroesophageal reflux-induced mucosal injury. To further explore the effect of IL-1 beta, we stimulated FEF3 cells and patient-derived fibroblasts with IL-1 beta for 24 hours, and then ran PCRs to quantify the expression of known extracellular matrix remodeling markers. So ACTA2 for alpha smooth muscle actin, fibronectin 1, and collagen 1A1. We also quantified the expression of collagen 8A1 based on the results from our fetal lamb model for esophageal stretching. In our lamb model, a partial esophagectomy was performed and the esophagus was stretched by joining both hands. The esophagus was then harvested at different time points, and RNA sequencing and proteomic study were performed on the tissue. Results from the omics analysis showed an increase in collagen 8A1 at every time point. Now back to fibroblast stimulation. These graphs show the relative mRNA expression of ACTA2, fibronectin 1, collagen 1A1, and collagen 8A1 in all 3 cell lines after IL-1 beta stimulation. What we can see is that the expression of the 3 extracellular matrix remodeling markers fluctuates a lot in all 3 cell lines, with IL-1 beta sometimes increasing and sometimes decreasing the expression of these markers. This might suggest that IL-1 beta regulates rather than just stimulates the expression of these markers. You can also see that each cell line tends to react differently to IL-1 beta stimulation. Collagen 8A1 was the only gene whose expression was consistently increased after IL-1 beta stimulation. We then performed a contraction assay on FEF3 cells using IL-1 beta. Contraction assays are functional tests for fibroblast activation, where the cells are embedded in a collagen matrix. Then the matrix is scraped off from the well and will shrink as the fibroblast contracts. You can see on the left-hand side a picture of the matrix shrinking throughout time. And the graph on the right-hand side shows you the change in the surface area of the matrix at those same time points. And you can see that IL-1 beta significantly increases fibroblast contraction between 1 and 6 hours after the release. In conclusion, these are preliminary data suggesting that stretched fibroblasts auto-activate via IL-1 beta expression. In turn, IL-1 beta may regulate extracellular matrix protein synthesis. Collagen 8A1 has been shown to activate the TGF-beta pathway in vascular diseases, but it raises the question of its precise role in esophageal fibrosis. Thank you.
We have time for one question.
Hi, my name's Carly Calabrese. I'm a postdoctoral research fellow at Stanford. You utilized 1 Hertz, and we are curious as to why you did that, since Hertz is a cycle per second, so it measures the circumferential force instead of longitudinal.
For the tension, yes, I'm sorry, and that's a typo that I didn't correct before. It was actually for 4 hours, and there's no, like, it's not a cyclic tension, it's a continuous one, so there's no hertz. I'm sorry about that.
Okay, thank you.
Sure.
Thank you so much.
Our next talk is titled Conversion of Omega-6 to Omega-3 Fatty Acids Protects Against Liver Injury in a Murine Model of Parental Nutrition-Induced Hepatosteatosis. Our presenter is Dr. Sarah Wang, Research Fellow at Boston Children's Hospital.
Good afternoon, everyone. My name is Sarah Wang. Thank you so much for the opportunity to present our work. We have no disclosures. Surgical patients on parenteral nutrition are at risk of developing liver disease, the pathophysiology which is multifactorial. It's important to understand the mechanisms of how PN can lead to liver injury, as it can lead to the development of improved nutritional therapy. Surgical patients may require long-term PN for a variety of reasons, including, for example, short gout syndrome. And PN dependence, we know, long-term increases the risk of PN-associated liver disease. And histologically, this can present with cholestasis, ranging to cirrhosis. Clinically, these patients can present with jaundice, growth delay, and liver failure, sometimes necessitating liver transplantation. We know that the intravenous lipid emulsion component of PN is associated with liver toxicity. And previous research has demonstrated high levels of phytosterols, which are plant cholesterols, and low levels of alpha-tocopherol, an antioxidant, it's vitamin E, has been associated with liver injury. However, the role of pro-inflammatory omega-6 fatty acids is lesser known. It's important to emphasize that humans are unable to endogenously synthesize omega-3 fatty acids, which have innate anti-inflammatory properties, so we must intake this through our diet alone. FAT1 transgenic mice express the omega-3 fatty acid desaturase gene. This enzyme helps to convert omega-6 to omega-3 fatty acids. The ratio of omega-6 to omega-3 fatty acids is different between wild-type mice and FAT1 mice. It's approximately 30 to 1.7. We therefore hypothesized that in a murine model of PN-induced hepatosteatosis, FAT1 mice will have reduced severity of liver disease compared to C57 mice when administered identical intravenous lipid emulsions that we commonly see in clinical practice. And this was measured by the degree of liver steatosis on histology. We utilize a well-established model of PN-associated liver disease. Mice are randomized either to a chow-fed control group or a parenteral nutrition group. In the PN group, mice are fed an ad libitum high-carbohydrate PN equivalent solution, and additionally, they're injected with intravenous saline as a control, SMOF or intralipid every other day until day 19, when all animals are euthanized for tissue collection. We observed no statistically significant differences between the weight trends between all of the groups, other than what is physiologically apparent between wild types and FAT1 mice at the baseline. In addition, parenteral nutrition consumption was documented, and we observed no statistically significant differences in PN consumption between the groups as well. Therefore, we can conclude that all animals involved in this experiment received identical nutrient intake. Free fatty acid analysis was conducted in these animals. FAT1 mice had lower omega-6 to omega-3 fatty acid concentrations compared to their wild-type counterparts across all groups, both chow-fed as well as all of the parenteral nutrition groups. And it is from this that we can conclude FAT1 mice model does in fact work in converting omega-6 fatty acids to omega-3 fatty acids. Liver histology was measured, and on H&E stain, lipid droplets appear as small white circular spaces demonstrated by the arrow. And FAT1 mice have fewer such areas compared to the wild-type counterparts that are similarly treated. This was particularly apparent for all of the parenteral nutrition groups. In addition, oil red O stain was conducted, and this is a specialty histology stain in which fatty deposition is illustrated as orange to red in coloring. An example shown here. FAT1 mice had fewer such areas compared to their wild-type counterparts, and this was again particularly apparent for the PN groups, notably the PN plus saline, as well as the PN plus intralipid groups. Liver triglyceride was quantified using an ELISA assay. FAT1 mice had lower liver triglyceride concentrations compared to C57 mice for the PN plus saline and the PN plus intralipid groups, and this approached statistical significance for the PN plus SMOF lipid group. In conclusion, in our mirroring model of PN-induced steatosis, FAT1 mice had reduced fatty liver disease in comparison to C57 mice when administered identical intravenous lipid emulsions. It is from this that we conclude the conversion of omega-6 fatty acids to omega-3 fatty acids confers hepatoprotective properties. Further research on the mechanisms behind how this occurs should be conducted in order to improve outcomes for our surgical patients who require long-term parenteral nutrition. I'd like to thank my research team, especially to my mentor, Dr. Mark Puter, sitting in the back there. Couldn't have done this work without our entire team, and I'm happy to take any questions at this time.
We do have time for one question.
That was a great talk. One question I had is what future next steps do you guys have planned now with the findings that you have?
Yeah, definitely. This project, although it has sort of reached a conclusion of our initial hypothesis, I think there's always more that we can do with the tissue we've collected. For example, looking at inflammatory markers and so on. So that's an area of active investigation in our lab.
Thanks.
Thank you.
Thank you so much. Our next speaker is Dr. Morgan Brown, who will be presenting the role of MEG3 in promoting proliferation and clonogenicity in hepatoblastoma.
Thank you so much for the opportunity to present our work today. Oh, we have no disclosures. Hepatoblastoma is the most common primary liver tumor in children. 20% of patients with hepatoblastoma present with metastatic disease, most commonly in the lung. Children with metastatic disease have lower event-free survival rates, and their long-term survival rates are as low as 25%. With this in mind, to better study this disease, our lab previously developed a metastatic hepatoblastoma cell line. Luciferase-positive cells from the human hepatoblastoma cell line, HUH6, were injected using tail vein injections, and IVUS imaging was used to detect bioluminescence and monitor for formation of lung metastasis. Metastatic tumors were harvested, dissociated, and plated in culture to derive the metastatic hepatoblastoma cell line HLM1. These cells were again injected into the tail vein of an animal, and the same procedure was followed to generate HLM2 cells. Previous work has shown that the HLM2 cells have increased proliferation, clonogenicity, migration, and invasion compared to HUH6 cells. So we wanted to investigate if there were any features of the HLM2 cells that are making them more metastatic that could be potential targets for intervention. We performed RNA sequencing comparing our HLM2 and HUH6 human hepatoblastoma cell lines, and specifically focused on the differentially expressed long non-coding RNAs between the cell lines. These are RNAs that are greater than 200 nucleotides and that are not translated into protein, and they function as epigenetic regulators. They can act as oncogenes or tumor suppressors, depending on the context. Of the differentially expressed long non-coding RNAs, we chose maternally expressed gene 3, or MEG3, as our long non-coding RNA of interest, as it was upregulated in HLM2 cells compared to HUH6 cells. You can see its structure featured here with 3 conserved motifs, M1, M2, and M3, that are found in all isoforms. In other cancers, MEG3 has been previously shown to act as a tumor suppressor where it is downregulated and plays a role in migration and invasion. Notably, some long non-coding RNAs have been shown to play dual roles as oncogenes or tumor suppressors depending on the cancer. Consistent with our RNA sequencing findings, we previously found that MEG3 was upregulated in our HLM2 cell line compared to our HUH6 cell line, in our HLM2 orthotopic tumors compared to HUH6 tumors, and in human hepatoblastoma tumor samples compared to normal liver. So from these findings, we hypothesized that MEG3 knockdown would reverse the metastatic phenotype of HLM2 cells with respect to clonogenicity and motility through colony formation, migration, and invasion. We employed small interfering RNA, or siRNA, to knock down MEG3. HLM2 cells were transfected for 12 hours with 20 nanomolar of control siRNA, or Si negative, or siRNA directed towards MEG3, or siMEG3. Downregulation of MEG3 was confirmed with quantitative PCR. Clonogenic potential was assessed using a colony formation assay, where HLM2 cells transfected with Si negative or siMEG3 were plated in culture, and after 11 days, colonies were stained and counted. We assessed migration and invasion of cells treated with Si-negative control or siMEG3 using transwell inserts coated with collagen to assess migration and collagen and Matrigel to assess invasion. Quantitative real-time PCR was used to confirm target engagement. Here you can see a significant decrease in MEG3 abundance with each of our siMEG3 components compared to the Si-negative control. Treatment with siMEG3 showed significantly decreased colony formation compared to Si-negative control with representative images shown below. Treatment with siMEG3 was associated with significantly decreased migration and invasion compared to Si-negative control. So in summary, MEG3 knockdown results in decreased clonogenic potential and decreased motility of HLM2 cells. These results provide evidence that MEG3 plays a role in the metastatic phenotype of hepatoblastoma and support further exploration of this. Our future experiments will focus on delineating the mechanism by which MEG3 plays a role in metastatic hepatoblastoma. I'd like to thank Dr. Byerly and the Byerly Lab, and I'd be happy to take any questions.
Hey, Andrew Murphy from Memphis, Tennessee. I was wondering if you had any insights on what the mechanism of MEG3 loss was in the model, since it's an imprinted gene and, well, an imprinted RNA, and if there were any more global disruptions in imprinting that you identified in the model.
Not that we've identified so far, but we are looking at a couple of targets related to motility that we're exploring further as a mechanism, so hopefully more to come on that. Thank you.
Our next talk is titled Delineating the Production of Matrosome Proteins by Ovarian Stromal Cells for a Bioprosthetic and Transplantable Ovary. Presenting is Dr. Christopher McCauley from Lurie Children's Hospital.
Thank you for the opportunity to present. We have no relevant disclosures. Approximately 16,000 children are diagnosed with cancer in the United States each year, with 40 to 60% of these estimated to be at some risk of gonadotoxicity as a result of their treatment. 80% of adult survivors of childhood cancer, however, indicate a possible or definitive desire for biological children. Opportunities for fertility preservation can be time limited in the setting of a new cancer diagnosis, and the only fertility preservation option for prepubertal girls is ovarian tissue cryopreservation, or OTC. OTC requires laparoscopic unilateral oophorectomy, tissue processing, and storage of ovarian cortical strips, which possess the primordial follicles or potential eggs for long-term storage. The goal is eventual autotransplantation of these cortical strips for the restoration of hormone and fertility function later in life. However, ovarian tissue transplantation is not an option for up to 1 in 6 patients in our series due to the risk of reimplantation of malignant cells or disease-specific issues. An alternative solution may be a bioprosthetic ovary. Our group utilized a 3D printed matrix combined with green fluorescent protein, GFP positive follicles, to restore fertility in previously oophorectomized sterile mice. This resulted in a GFP positive pup, and these pups maintained GFP positivity and fertility, ultimately giving rise to a second generation of GFP positive pups. We aim for our next bioprosthetic ovary to have improved longevity by decreasing activation of primordial follicles, improving structural support, and for interstitial cells and the matrosome to support neovascularization and increase follicular stability. The matrosome consists of 1,000 ECM proteins and supports several critical ovarian follicular processes such as neovascularization and hormone signaling. We also know that when we change the matrosome composure of hydrogels, ovarian follicles grow at different rates. The hypothesis of this work is that stromal cells, isolated and cultured from the ovarian cortex or near the surface of the ovary, and medulla or near the middle of the ovary, maintain proteomic memory and express matrosome proteins that reflect the compartment from which they came. Ovaries were sliced in the sagittal and axial plane, decellularized, and bottom-up shotgun proteomics was utilized to determine the presence and relative abundance of matrosome proteins by depth within the ovary. Interstitial cells were then mechanically and enzymatically isolated from the first slice or cortical interstitial cells and from the fourth slice or medullary interstitial cells and cultured. ICC was then utilized to probe for matrosome proteins. In terms of our proteomic results, we identified 291 unique matrosome proteins. 65 of these proteins were differentially expressed between cortex and medulla. We then selected several of these differentially expressed proteins to interrogate and culture. As you move from left to right in this heat map, you are progressing deeper into the ovary, from cortex to medulla. We utilize our proteomic data to select our matrosome proteins for interrogation. Here, we see that two proteins that are cortex predominant, 2 that are equal between compartments, and 2 that are medulla predominant. We first characterized growth patterns of isolated interstitial cells. Both cortical and medullary cells were successfully isolated and grow rapidly. Cortex doubling time was 18 hours and medullary doubling time was 27 hours. ICC was then performed on isolated stromal cells of cortical and medullary origin. High-powered images of nearly an entire well from a 24-well plate were collected from 3 bioreps and 3 tech reps. Intensity per cell was compared. The left pane are cortical cells and the right pane are medullary cells, stained for FRAGE1, a matrosome protein. FRAGE1, a cortex-predominant protein, was slightly more abundant in cortex, though not statistically significant. Vitronectin and TGF-beta proteins where cortex on the proteomics are equal between the cortex and medullary compartments were slightly more abundant in the medullary cells, with TGF-beta reaching statistical significance. And CST3, a medullary predominant protein, trended towards increased abundance in the medulla, though not statistically significant. In conclusion, we have successfully isolated and cultured stromal cells from the cortical and medullary components of the ovary. These cell populations expand rapidly and produce matrosome proteins from multiple matrosome categories. Within the limits of ICC protein quantification, it is not yet clear if cortical and medullary cells maintain compartmental proteomic memory. Next steps would include Western quantification, qPCR, and/or RNA sequencing to further interrogate our hypothesis. I'd like to thank the members of the FHPR team, and I'd be happy to take any questions.
We do have time for one question. And if there are no questions at this time, we'll move on.
Yeah,
Thank
thank
you
you.
so much.
Thanks.
Our next speaker is Dr. Leah Ott. And the title is Subcutaneous Adipose Tissue-Derived Neural Stem Cells, a novel autologous cell therapy to bridge the gap in the treatment of neurogenesis.
Great. Good afternoon, and thank you for the opportunity to present. We have the following disclosure regarding a provisional patent. Unlike the central nervous system, peripheral nerves do retain a limited capacity to repair themselves, but outcomes remain poor for large gap transection injuries, particularly those greater than 3 to 5 centimeters, with a minority of patients achieving appropriate sensory and motor recovery, particularly in the trauma population. To date, the gold standard treatment for these injuries is autologous nerve grafting, but there are multiple drawbacks to this approach, including limited appropriate donor nerves, as well as donor site morbidity. Artificial nerve grafts have been under development, but continue to have limited efficacy in these large gap injuries. Schwann cells are known to play a critical role in endogenous nerve repair and were thought to be appropriate seed cells initially to help boost the efficacy of these conduits. However, they're very difficult to propagate in culture, and thus instead, attention has turned to neural stem cell therapy for these injuries, given neural stem cell capacity to differentiate into Schwann-like cells, to promote myelination, to produce various growth factors, and also to drive angiogenesis. Our lab has previously shown that Schwann cells residing in subcutaneous adipose tissue can adopt neural stem cell properties in vitro. So with these red Schwann cells in this picture, then adopting the neural stem cell marker P75 in vitro. And similarly, when we performed quantitative PCR, we found that the Schwann cells in vitro downregulated mature Schwann cell markers such as myelin basic protein and myelin protein 0, and upregulated the neural stem cell markers Nestin and P75. They could be transplanted into the GI tract, under which conditions they then differentiated into neurons and glia, as seen by the co-staining of beta-tubulin 3, a neuronal marker, with the red neural stem cells. So then we were interested in applying these cells in a different peripheral nerve injury model outside the GI tract, and so found that we could deliver our adipose-derived neural stem cells via an artificial conduit to an injured peripheral nerve. We found that these cells could survive at postoperative day 56, as seen by the red fluorescence in the gross image, and that they could integrate with the host population of Schwann cells shown in green fluorescence. We found that these cells would not express the neuronal marker beta-tubulin 3, but rather, like the host, Schwann cells express the marker LGI1-ish mature Schwann cell marker. And so we were then interested in applying these cells in a biological conduit using decellularized nerves to see if they could be similarly applied and transplanted in a different modality that's more appropriate for clinical translation. So utilizing either dual neuronal and glia reporter BAT53 TDT PLP1 GFP or the pan neurocrest reporter WNT1 TDT, we took subcutaneous adipose tissue from mice, enzymatically digested it, passed it over a 70 micrometer filter to isolate the nerve fiber bundles, and then propagated them in culture. We then performed sciatic nerve injury surgery in mice, in which the sciatic nerve is sharply transected at the mid-thigh and immediately repaired with a 5 millimeter gap. And then to create decellularized conduits, we took cadaveric nerves and decellularized them using previously published protocols, enzymatically digesting them, and then directly cultured our subcutaneous adipose-derived neural stem cells on these conduits. We then perform walking track analysis, in which the mouse's hind feet are painted with India ink, and we look at paw length and toe spread that can then be quantified to measure sciatic functional index and measure of sciatic nerve dysfunction, with 0 being normal and minus 100 being complete dysfunction. We also looked at gastrocnemius muscle weight, and using immunohistochemistry, looked at neural stem cell survival and integration. And so we can see that our red fluorescent subcutaneous adipose tissue-derived Schwann cells are capable of becoming clusters of neural stem cells in vitro, and that these cells could be successfully cultured directly onto a decellularized nerve, and that after 10 days or 8 days in culture, that they could propagate along the decellularized nerve and cover greater surface area. Next, we performed our sciatic nerve injury surgery and found at postoperative day 56 that our repair was intact, and that using fluorescent microscopy, we could see that our red fluorescent adipose-derived neural stem cells persisted and integrated with the host green fluorescent Schwann cell population. also shown here at greater magnification. Next, when we utilized a dual glial neural reporter, we found that our Schwann cells, or our adipose-derived neural stem cells, only expressed PLP1 GFP, suggesting that they're undergoing Schwann-like cell differentiation, but did not express mature neuronal markers after transplantation. And when we looked at gastrocnemius muscle weight as compared to mice who had no repair, both an empty decellularized nerve and a decellularized nerve with our neural stem cells led to greater muscle weight, but we saw no statistically significant difference in sciatic functional index. And so, in conclusion, we find that these cells may represent a novel source of autologous neural stem cell therapy for such injuries that could be harvested through a minimally invasive means, such as through a liposuction specimen, and that we can use them apply them in the peripheral nervous system to undergo Schwann-like cell differentiation to promote recovery. But the exact conditions to lead to optimal recovery still remain to be seen. I'd like to thank the Goldstein Lab, particularly my mentor, Dr. Goldstein, as well as our sources of funding, and I'd be happy to take any questions.
James Dunn, Stanford University. Very nice work. In the past, we've done these experiments with what's called the SKP cells. They're also skin-derived neuroprogenitor cells. Are these similar to those, or are they separate? It looks like you just culture them under a certain culture medium that selects for them. Is that correct?
Yes, so we basically culture these nerve fiber bundles that exist in adipose tissue in a neuroproliferation media that has basically various like neural progenitor factors, and that we've shown in other experiments that like within the nerve fiber bundle you have both axons and Schwann cells, and because the axons are cut off from the cell body in the spinal cord, only the Schwann cells are those that are persisting, and then we believe they're undergoing dedifferentiation into a neural stem cell phenotype. But it's certainly possible too that in the skin we have some questions that these neuronal differentiation properties, at least in adipose, were thought to be from mesenchymal stem cells or mesenchymal stromal cells initially. And we wonder if perhaps that in these prior experiments there was a contaminating population of Schwann cells that actually underlies that phenomenon.
Thank you.
Our next talk is titled Repeated Lengthening Utilizing Distraction and Pterogenesis, a Porcine Model. Presenting is Dr. Pamela Emengo, a pediatric surgery postdoctoral fellow at Stanford University.
Hi, everyone. My name is Pamela Emengo. Thank you for the opportunity to present today. Dr. James Dunn is the creator of the springs and owns a patent and a company to make them. The springs we use in our experiment were made by our lab and not the company. Spring-mediated distraction anaerogenesis uses springs to stretch the intestine and generate new intestinal length. Pictured here is a figure from our previous work showing that we take a spring, compress it down, place it into an intestine, and then over time it'll grow new intestine. Each spring can lengthen the intestine up to 3 to 4 centimeters. While this is important, it may not be enough to cure short bowel syndrome in every patient. That's why our lab has researched different ways to expedite growth. We've previously researched in small and large animal studies placing multiple springs at one surgery. We've also taken a look at repeating the distraction process in a small animal model. But before we move on to placing springs in humans, we wanted to test the feasibility of repeated distraction in a large animal model. To test the ability to repeat distraction, we perform a total of 3 surgeries on one pig. In the first surgery, we take the segment of jejunum and place a 5 Newton-meter force spring and plicate the spring in place and use a suture to demarcate a section of control intestine to serve as our internal control. We wait up to 21 days before we start our second surgery. In the second surgery, we look at the area of new growth, and after we measure, we remove the spring and place a new spring of 10 Newton-meter springs of Newton-meter force. We use a stronger spring to account for the expected muscle hypertrophy that we found in previous studies. And then we wait a period of 7 more days before we move on to our third surgery and euthanize the animal and collect our samples. In each surgery, we are measuring the spring segment length and the control segment length and calculate the percent change. Now I want to show you some photos representative from our surgeries. Keep in mind that the black arrows in each photo represent our spring segment, and our white arrows represent our control segments. So here you can see in our first surgery, we place a compressed spring, and we look at our control segment. As we move on to our second surgery, we see that the area of previously compressed spring has lengthened while our control segment stays the same. In that same surgery, we take out the old spring and we place a new spring into that previously lengthened segment. And then in our final surgery, we euthanize the animal and harvest all the specimens. In total, we performed 6 surgeries on 6 pigs. Each spring, we noticed had an increased intestinal length. So here's a graph showing the % change in length of the intestinal segment. You can see that in spring 1, the change in length is about double compared to the original. And there was no difference between change in spring 1 and spring 2. We also took a look at our histology and quantified it, and we saw that there was increased crypt depth, mucosal thickness, muscularis thickness, and outer perimeter of our spring segments when we compare it to the control. Here are some photos from our H&E sections, and you can see our control segment. You can see all 4 layers of the bowel wall, but when you take a look at our spring segment, you notice the hypertrophy of the muscularis propria and our crypts very well. We also looked at immunohistostaining to further quantify our histology. When we looked at myosin heavy chain 11, which is a marker of smooth muscle cell, we noticed that even though our muscle had increased, it maintained normal orientation. We also stained for olfactin median 4, which is a marker of intestinal stem cells. And you can see that in our spring segment, we had increased expression of olfactin median 4 when compared to our controls. Overall, you can see that repeated distraction is feasible in our large animal models, and our next steps are to implement distraction in our upcoming clinical trials. These are our references, and thank you everyone for listening, and thank you Dr. Dunn for all your mentorship and support.
We do have time for questions.
Thanks. Congratulations on some amazing work that looks like it may be transformative. Do you have any idea what the motility is of these segments?
So we've performed several different surgeries in the past looking at long-term function of our spring segments. And after a period of time, once the spring is removed, the intestine returns back to normal. One of the studies we've done in pigs, we looked at the function 6 months out after surgery, and the histological changes had normalized.
I'm sorry, when you say returns back to normal size or returns back to normal function?
Both. The histological changes return back to normal and the function. So all of our pigs throughout the surgeries are eating, playing, doing normal activities, even when the spring is in place.
Cool.
Are there any other audience questions?
If not, I have a question. That was a great talk, super interesting. My question is, when you saw the changes in the cell types, the muscle layers and things like that, did you see the changes throughout the entire segment, or were there greater changes at the area where the spring's kind of pushing on the intestine?
So we take our histology from the actual spring segment itself. So the changes that I showed in the previous figures are representative throughout the entire segment we've taken. We also looked at the control segment for changes, and the changes we see in the spring segment are not in the control. But we are looking into future work, seeing how our control segment compares to just normal unmanipulated bowel.
Thank you.
And I'll follow that with a question about the segment of the intestine in which the plication sutures are applied. Have you undertaken histologic investigation of those segments, and are there any concerns for stricture or narrowing? Are those plication sutures resorbable, for example? What can you tell us about those segments of bowel?
So in our animal models, we use a non-absorbable suture to perform our plications. But in the upcoming clinical trials, we'll be using absorbable because we don't want to run the risk of any stricture or long-term consequences of suture.
Thank you so much. Our next speaker is Dr. Celebres, and the title is The Effect of Distraction Enterogenesis on Mechanosensitive Pathways in the Marine Cecum.
Thank you for providing me the opportunity to speak to you all today. Here are our disclosures. As mentioned previously, my mentor, Dr. James Dunn, has a couple of patents and is the co-founder of Eclipse Regenesis, a company that manufactures springs for clinical use. The springs that we used in this study were not supplied by Eclipse. It is our aim to improve the longevity of life in children with short bowel syndrome and intestinal failure. On average, 35 out of 100,000 newborns are impacted by short bowel syndrome, which is a high morbidity and mortality rate. A number of these children require parenteral nutrition, which is associated with a host of complications, including liver failure, bloodstream infections, thrombosis, among others. Necrotizing enterocolitis, volvulus, Hirschsprung disease, among other conditions, can result in short bowel syndrome, which is the leading cause of intestinal failure in children. Intestinal failure is often managed with a combination of parenteral nutrition, medications including GLP-2 analogs such as Gattex, and distraction enterogenesis, which my colleague Pamela just discussed. Distraction enterogenesis has shown promise in animal models by applying mechanical force to generate intestinal tissue. We had previously published the results of a retrospective chart review that revealed that children with short bowel syndrome that have their entire colon intact are able to wean from parenteral nutrition at a quicker rate than children that had had at least a partial colectomy previously. This demonstrates the importance of preserving the colon in these children. If we can preserve or even lengthen the colon, we may be able to improve outcomes. Therefore, in this study, we inserted nitinol springs, both compressed and uncompressed, into the blind end of the cecum in mice. Here's a photo of the blind end of the cecum, and here's a photo of the compressed spring after we placed it into the cecum. Over the course of 7 days, the compressed spring resulted in distraction enterogenesis, or lengthening of the cecum. Here's a photo of the cecum after the spring had resulted in lengthening and generation of intestinal tissue. We measured the spring segments at time of placement on postoperative day 0 and also at time of planned euthanasia on postoperative day 7. We determined differences between the mice that had compressed springs and the mice that had uncompressed springs using t-tests. We then performed RNA sequencing to evaluate the gene expression changes in the tissue. Overall, we inserted compressed springs into 5 mice and uncompressed springs into 4 mice. We found that the compressed springs resulted in 66 to 83% increase in cecal length over the course of 7 days. At the time of placement, the spring segment measured 6 millimeters, and at the time of postoperative day 7, that segment had grown by 4 to 5 millimeters. This was statistically significantly increased compared to the lack of lengthening we noted in the uncompressed spring mice. Subsequent gene expression analysis revealed that any acute changes that may have resulted from the distraction enterogenesis had normalized by postoperative day 7. However, we wanted to look at this further, so we lessened the scale to determine if there was any gene upregulation at this time point. And we did find that, in fact, there was some gene upregulation in the lengthened tissue or in the mice that had compressed springs inserted. We specifically focused on mechanosensitive genes, PIZO1 and PIZO2, and their downstream effectors, genes involved in cell proliferation, growth, and differentiation. Again, noting that the mice with compressed springs and subsequent cecal lengthening had some gene upregulation. Overall, we have found that distraction enterogenesis is safe. It promotes lengthening and can regenerate cecal tissue. With use of springs, we can generate tissue that looks very similar from a genetic standpoint to tissue that was not successfully lengthened with uncompressed spring insertion. And although there may be acute changes at play that were not realized in this study, there is no sustained substantial upregulation of proliferative pathways. Next, we aim to identify the various pathways activated in response to mechanical forces at shorter time points, including postoperative day 1. Thank you very much. I'm happy to take any questions.
we do have time for questions from the audience. And if there are none, I will take advantage of the podium and ask whether you've given consideration to single cell analysis as something that might elucidate some of the mechanotransductive pathways that are almost surely at play.
Yes, most certainly. That is one of our next steps, and I'll talk a little bit more about some more gene sequencing analysis that we have done in the next talk. But yes, next we do plan to look at single cell and further study these mechanosensitive pathways that are occurring in this process.
Okay, if there's no more questions, we'll introduce your next presentation. So the next presentation is titled Mechanical Force-Driven Epithelial Growth, YAP Activation, and ISC Stemness in Non-Regenerative Mechanisms.
Again, thank you for providing me the opportunity to present our work today. We have no disclosures relevant to this particular study. It is our goal to identify novel therapies in the management of short bowel syndrome. YES-associated protein, or YAP, is a mechanosensitive transcriptional coactivator that is heavily involved in determining cell fate. We want to determine whether YAP is involved in growth pathways instead of an injury or inflammatory state. Specifically, we set out to delineate a mechanical force-induced epithelial growth signaling pathway. But why in the big picture does all of this matter to us? Because children with short bowel syndrome often require intestinal lengthening procedures and as a last resort, small bowel transplant. These procedures are associated with a great deal of morbidity and mortality. What if we could identify less invasive therapies that could better manage children with short bowel syndrome and not subject them to as much risk? Nuclear YAP is important in the self-renewal of intestinal stem cells, proliferation, and growth. There are a number of pathways involved in the translocation of YAP from the cytosol, where it is inactive, to the nucleus, where it can serve as a transcriptional coactivator. The Hippo pathway is just one of these pathways. When Hippo is off, YAP is not phosphorylated, and it can translocate to the nucleus and serve its role as a transcriptional coactivator. We know that YAP is activated in several pathological states, including uncontrolled cell proliferation, cell differentiation, and post-injury regenerative and inflammatory states. But what about its role in force-induced intestinal epithelial growth? This has not been well defined. In order to answer this question, we used a partial obstruction model. We placed a 5mm rubber band around the terminal ileum of mice and subsequently harvested their dilated proximal bowel on postoperative days 1 through 7. We compared this bowel with that of sham mice. For the sham mice, we placed the rubber band around their terminal ileum, but subsequently removed it prior to abdominal closure, presumably resulting in some inflammation, but not in a force-induced epithelial growth model. We harvested the bowel from the sham mice on postoperative day 3. We then performed immunofluorescence to evaluate intestinal stem cells, YAP, and proliferation. We used an image-based model to track the nuclear translocation of YAP. And we performed RNA sequencing to identify the genes involved in this process. Overall, on postoperative day 3, we noted that the tissue was maximally stretched out. At this time point, the lumen radius was increased, the muscle thickness was reduced, and the crypt density was reduced, suggesting that the tissue was experiencing peak mechanical force. Subsequent immunofluorescence showed us where the intestinal stem cells are located within the crypts. You can see them here outlined with the white dotted lines. You can see that the YAP in the intestinal stem cells was not as highly expressed in the sham mice as compared to the obstructed mice on postoperative day 3. YAP intensity increased over time, with the maximal nuclear to cytosolic ratio occurring on postoperative day 3. Then, by postoperative day 5, we noted that the percent crypt fission rate had maximized. Here with the histology, you can identify the process of crypt fissuring. We utilized the immunofluorescent marker Ki67 to identify that proliferation was elevated in the epithelial and muscle layers at this time point. We also noted that on postoperative day 5, the amount of intestinal stem cells per crypt was at a maximum. And you can see that here with the immunofluorescent marker OLFM4. Subsequent gene expression profiling revealed that the mechanosensitive and proliferative pathways were upregulated at postoperative day 5 compared to 3, specifically looking at the intestinal stem cell stemness and the Wnt signaling pathway. This is in contrast to the inflammatory pathways, which were downregulated on postoperative day 5 compared to 3, which are noted on the bottom of the screen. We also noted that the YAP downstream targets were upregulated on postoperative days 3 and 5. This correlated with the Wnt non-canonical pathways. We noted that EGFR pathways were upregulated in the sham mice, but not in the obstructed model. Overall, we have identified that mechanical force does induce epithelial growth and intestinal stem cell expansion. And we have determined a previously unexplored role of YAP in intestinal growth. This is exciting because we may be able to better treat epithelial impairments, such as short bowel syndrome. Next, we plan to determine more specific spatiotemporal, YAP-dependent, and independent growth-promoting targets in single-cell resolution. Again, thank you so much for your attention. I'm happy to take any questions.
Hi, I'm Linda Lee from Mount Sinai. That was a great talk. It went all over my head, but bear with me. I have a question based on your model and the temporal time that it associated with it. And I'm just thinking, if you're essentially putting stretch on an intestine, and the other time that I can think of we put stretch on bowel or GI tract is with long segment esophageal atresia. So I'm wondering, do you think based on your findings that if we apply that to a long gap esophageal atresia that maybe we ought to be going back sooner? Because I'm thinking about sometimes when we do those stretch techniques, we're going back like months intervals or week-long intervals, but do you think things stabilize earlier than we think?
I wouldn't make that conclusion based off of the results that we have at this time point. I think it's hard to make that clinical correlation given its different tissue. We have tried to make some clinical connections in regard to the genes activated in healthy dilated colon from Hirschsprung's patients and have noted a similarity between the genes activated in those patients compared to the genes activated in our partial obstruction model. But I wouldn't go as far to say that it's the same thing to apply everything that we've found to the esophagus. And I think there's a lot of mechanisms at play that we're still kind of trying to figure out before we can make such conclusions.
Thanks so much.
Yeah.
And Joe Church from Michigan, that's really fascinating work. I think, especially with your back-to-back presentations, you kind of bring up, when you talk about this growth or stretch, these mechanical forces, you can have radial growth or longitudinal growth, one of which can sometimes be maladaptive, the other which sounds like it's your overarching goal. And how YAP factors into this to me is totally fascinating. I'm wondering, putting the two together, if you have looked into or are going to look into perhaps the role of YAP or related signaling pathways with your spring-induced bowel lengthening.
Yes, absolutely. Yes, that will be a next step for sure. I think this is, a way that we can look at circumferential force, which is different than longitudinal. And it's exciting to think that we'll eventually look at these pathways in after spring insertion as well. Thank you for your question.
Yeah, thank you.
I'll add that as our labs are collaborating and exploring the role of lengthening of the esophagus with distraction enterogenesis, we're paying increased attention to the role of fibrosis and fibrotic change in stretch tissue. Do you have any insights into how fibrosis may play a role in lengthening in the cecum and the small bowel, and how you might explore potential ways to modulate that effective lengthening in the future?
Yeah, so fibrosis is certainly can be a result of spring insertion, and you do have to adjust the strength of the spring in order to kind of account for that, which Pamela touched on. But I think there's a lot of mechanisms at play that we need to kind of further delineate and determine, which cells are involved at which time points and whether there's a difference depending on the location of the cell in regard to where it is in the crypt, those sorts of things. So we kind of have to figure all that out to kind of figure out how to apply that.
Thank you so much.
Thank you.
And with that, this concludes our session. We will now have our first break of the afternoon. Please stop by and thank our vendors for their support of the conference and check out their offerings. Following the break, we will start our second series of scientific sessions, 13 through 15, starting at 4 p.m. Following the second scientific sessions, you will have some free time, and then we invite everyone to attend the President's Cocktail Reception from 6 to 7 in the Park Mount Royale, followed by the President's Dinner, scientific and individual award presentations and dancing. Thank you.